Um,
Okay. So maybe we should start right now.
yeah.
Okay, so you're hearing me well. Okay, good. So good morning, everyone. It's my pleasure to start our first EvoFunPath seminar of this new year by introducing our speaker, Dr. Znaidi, who is an assistant professor at Memorial University in Newfoundland and Labrador. So Dr. Znaidi is also affiliated to the International Network of Pasteur Institute. Dr. Znaidi did his PhD with Dr. Martine Raymond at University of Montreal. And he was one of the first using chip technique in Candida albicans to characterize the function of different transcription factors. Especially many of them, that are involved in antifungal resistance. That include, for example, TAC1, UPC2, for those who's working in Candida albicans. So during his PhD, he also contributed to the annotation of the genome of Candida albicans, specifically transcription factor of the zinc cluster family. For those who work on Candida, those proteins were annotated by Dr. Znaidi using the ZCF acronym, followed by unique digit or numerical digit. So after his PhD, Dr. Znaidi moved to Pasteur Institute in Paris for a postdoc in Dr. Christophe Denfer group, where he spent almost 6 years working on genetic determinism of fungal virulence using different omics approaches. In 2016, he joined Pasteur Institute in Tunisia as an independent scientist and made many significant contributions related to how bacteria and fungi interactions impact the human host. A few months ago, Dr. Znaidi moved back to Canada, precisely to Memorial University in St. John's, Newfoundland, as an assistant professor. So today, in his talk, Dr. Znaidi will address one of the key and long-standing questions related to the biology of an opportunistic pathogen or fungal pathogen, which is how these fungi switch from a commensal to a pathogenic state and vice versa. Thank you, Dr. Znaidi, for accepting our invitation. And before leaving you the stage, I just want to notice our training that Dr. Znaidi is perfectly bilingual, so feel free to ask questions in either French or English. You can also, after that, you can just ask directly questions if you have. You can also send them to me in the chat. I'll take care of asking them to Dr. Znaidi. So, Dr. Znaidi, thank you, and the screen is yours.
Merci, Adnan. Yeah. Thank you very much for this introduction. I really appreciate always being connected to you, especially as a colleague and friend as well. And thanks, Marie-Hélène, for managing all of this talk since a couple of months ago. And I'm happy and excited to be here in this EvoFunPath network training program on the evolution of fungal pathogens. So do you hear me? Yes, okay, wonderful. So I'm gonna be giving really a summary of my work on how to basically decipher the transcriptional circuitries with two main technologies basically that are cornerstone in constructing networks. And I'm going to be providing an overview of what I've done before, and also a second part of my talk will be more dedicated to recent work that we've performed at Institut Pasteur in Tunis, and linking somehow the function of transcription factor to the ability of Candida albicans to interact with bacterial species, right? And so the I would be introducing my talk with a general concept about transcription regulation in eukaryotes. So it's based on the binding of sequence-specific DNA binding proteins called transcription factors upstream of their target genes, followed by recruitment of the RNA polymerase II holoenzyme to activate gene expression. So this way, transcription factors act as activators of gene expression. Transcription factors can also act as repressors of gene expression, and they do so by recruiting so-called, for example, general co-repressors. These can be, for example, the famous Groucho family of regulators such as TOP1 or transcription general co-repressor, or also TOP11 is Saccharomyces pombe, for instance, if we talk about yeast there. So this way, transcription factor can act as an activator of gene expression and also as a repressor of gene expression. And so the genome of the 3 yeast species I'm showing here, fish and yeast, Saccharomyces pombe, baker yeast, Saccharomyces cerevisiae, and Candida albicans, the opportunistic yeast, encode roughly the same proportion of transcription factors, going from 2 to 3.5%. And the function of most of these transcription factors is unknown. And so one can hypothesize that by identifying transcription factor targets and the biological circuitries under their control could help defining their function. Now, how to do that? By combining basically two functional genomics approaches, two technologies. One is called genome-wide expression combined to genome-wide location. By expression, we mean RNA-seq, and location, we mean ChIP-seq. And so these two technologies allow to construct, are sufficient basically to construct transcriptional networks, like those modeling the functional interactions occurring between transcription factors and target genes, and predict, based on that, predict their biological function and test it experimentally. And so here is the overview of the use of those two approaches to decipher regulatory circuitries. First, ChIP-seq, chromatin immunoprecipitation, combined to high throughput sequencing. And to do that, we usually take a transcription factor, that's square here. We tag with an epitope tag, for instance, triple HA, MEC, or TAP tag, tandem affinity purification. We treat the cells with formaldehyde to induce cross-links between the protein and DNA. We prepare total extracts. We shear DNA with sonication to get, on average, a fragment of about 200 base pairs, 150 base pairs, and we immunoprecipitate the transcription factor that bound to this DNA with an antibody that recognizes specifically the epitope tag. Then we reverse cross-links by heating to 65 degrees Celsius and treating with STS and purified DNA. And then we perform high throughput sequencing of those fragments, right? And we map on using a mapping algorithm to get those peaks indicative of binding of transcription factor upstream or downstream of a target gene. In parallel, we perform in the same condition the RNA-seq analysis. So we extract total RNA, we reverse transcribe it, and we make our libraries, and we do RNA-seq analysis basically. And we look at the differential gene expression, and we combine those two technologies and data from those two technologies to map the transcriptional circuitry. How this works by looking at those genes that are bound and upregulated or downregulated in the transcript profiling data. So this allows to build the networks So looking at the transcription factors that acts as a direct activator of some genes or direct repressors of the expression of some other genes. And this way we could go one, but transcription factor by transcription factor and looking at the whole transcriptional circuitry there. So this allows to model basically the regulatory interactions occurring between transcription factor and target genes. And then later on, we could come up with additional analysis based on that, such as motif discovery analysis to look at the enrichment of motifs that in upstream sequences that are bound by this transcription factor, so we know where this transcription factor will bind to. We perform gene ontology enrichment analysis to look at the functional categories of those genes that are enriched, and this would predict potential function of those transcription factors. And then, of course, testing experimentally the potential function in vitro or in vivo in animal models, in the case of pathogenic fungi, for instance. So let's focus on this yeast, Candida albicans. Candida albicans, you all may know that this is a part of us, basically. It's part of our microbiome, intestinal microbiome, but also vaginal microbiome. It's a polymorphic yeast, so it exists in a yeast form, but also in a hyphal or filamentous form. And this switch from the yeast to hyphal form is important for its capacity to invade tissues and cause disease. So it's an opportunistic yeast. Basically, it acts as somehow a member of the microbiota that colonizes the microbiome colonizes the gastrointestinal tract with bacteria, for example, but also under immunosuppressive conditions, it can cause disease. So that's why we are opportunistic yeast, basically. So it causes superficial and invasive candidiasis. This is the name of the disease caused by Candida species, infectious disease caused by Candida species. So it causes, among the superficial infections, we have the oral candidiasis. You see that these are patchy lesions on the oral cavity, yellowish a bit, indicative of Candida that is stuck to the mucosa, also called thrush, but also it can cause onychomycosis. This is the inflammation of the region surrounding the nail here that could be seen as well. Now, when the host is severely immunocompromised, Candida albicans can translocate from the gastrointestinal tract to the bloodstream. And we see the so-called invasive infections where we can detect Candida albicans there in the bloodstream surrounded by immune cells, for instance. So shown here the yeast form, but also the filamentous form here. This is called candidemia. So the presence of Candida albicans or Candida species in the bloodstream is called candidemia or systemic candidiasis. This allows Candida to reach additional organs, including the eyes, for example, to cause ocular candidiasis. So it's really it could cause devastating infections in severely immunocompromised patients, basically. And so Candida has a diploid genome consisting of 8 chromosome pairs with a size of approximately 15.5 megabases, including 6,200 ORFs approximately, exactly 6,198. And so no meiosis has been observed in Candida albicans so far. And following mating, what happens is that the so-called perisexual cycle to lose extra copies of the chromosomes. From 2N, we go to 4N, and then back to 2N through this perisexual cycle. It is characterized by a relatively unstable genome with many instances of loss of heterozygosity and aneuploidy events. But despite of that, the molecular genetics tools are available. We can do genetics with Candida albicans and create mutants. And currently, many biological resources and genetic resources are under extensive development, including deletion mutants and overexpression collection, etc. Now, the treatment of candidiasis relies on administration of 3 main classes of systemic antifungal drugs. including polines, for example, amphotericin B that binds to ergosterol, leading to disruption of the fungal membranes. Echinocandins, for example, caspofundrin that binds to beta-1,3 glucan synthetase, leading to disruption of the fungal cell wall. And azoles, which are widely used both orally and intravenously, and they bind to 14-alpha-linosterol demethylase, leading to inhibition of growth. But so actually, when treatment fails, and treatment can fail based on side effects, because fungi are like us, they are eukaryotic microorganisms. So when we develop a drug that targets fungi, you would expect it to can target also the host side. And so drug toxicity is frequent in antifungal drug discovery, and so that's why it's challenging to find new drugs. In addition, there is potential for drug resistance. So when treatment fails, limited treatment options are limited, basically. And so it's important and critical to identify new drug targets and develop new antifungal drugs. That's why it's important to understand the molecular determinants of Candida albicans' ability to act as a commensal, but also as a pathogen, and see how we can target Candida for those reasons. So I'm going to be talking more now about the crucial, virulence threat is the ability of Candida albicans to go from a yeast form to a filamentous or hyphal form. So the yeast form that is usually observed during dissemination and surface colonization can switch into a filamentous, a hyphal form, important for invasion and internalization by host cells. And this transition is regulated by an intricate circuitry of signal transduction pathways. A lot of kinases there, and that that are activated through the stimuli, environmental stimuli, including matrix embedment, increasing in temperature 37 degrees Celsius, low nitrogen, incubation in serum, or elevation of CO2, or variation in methionine or some sugar derivatives, but also variation in pH. So you have different environmental stimuli, regulators of this ability of Candida albicans to switch from yeast to hyphal form. And the whole process goes to downstream to activation of a transcription factors, including, for instance, FLO8, EFG1, TEC1. All of those are characterized in Candida albicans from the perspective of their regulation of the yeast to hyphal transition, and mainly the regulation of the so-called hyphal specific genes, right, that allow Candida to develop into a filamentous form. On the other hand, you have repressors of hyphal formation, including, for instance, NRG1, RFG1, that also recruit the top one co-repressor to repress, gene expression. So you have this equilibrium between the transcription activators and transcription repressors. Now, there are two additional transcription factors involved in this process called SFL1, SFL2. These are two heat shock factor type transcription regulators involved in this process. So SFL1 acts as a repressor of hyphal growth, whereas SFL2 acts as an activator of this process. They are actually structurally homologous, but antagonistic in terms of function, which is really attractive to see how those two transcription factors regulate, filamentous growth. Consistent with the function of SFL1 as a repressor of hyphal growth, deletion of SFL1 activates hyphal growth in a condition that maintains yeast form growth. Now, reintroduction of a wild type copy of SFL1 restores yeast form growth. On the other hand, in a medium that activates hyphal growth, for instance, Lee's medium at 37, you have in a wild type activation of the hyphal growth. Deletion of SFL2 disrupts hyphal development. Now, reintroduction of a wild-type copy of SFL2 restores hyphal growth. And both transcription factors are involved in virulence in vivo models of systemic infection and GI tract colonization followed by dissemination. So those two transcription factors are important for hyphal development, filamentous growth, but also important for virulence. And so the idea here is to look at how this process occurs in a molecular level. So we combine those two technologies, the ChIP-seq and transcriptomics, as well as bioinformatic analysis to decipher the regulatory circuitry of SFL1, SFL2. So here is the transcriptional circuitry of those two paralogs with antagonistic function, basically. So what you can see already is that SFL1, the repressor of hyphal growth, directly activates the expression of repressors of hyphal development that we talked about initially, NRG1, RFG1, and activates the expression of the so-called yeast form-associated genes. In the meantime, it directly represses the expression of activators of hyphal growth and directly represses the expression of SFL2, its antagonistic counterpart here. On the other hand, to basically maintain the yeast form growth, right? On the other hand, SFL2, following overexpression or temperature increase to 37, it directly upregulates the expression of activators of hyphal growth, directly represses the repressors of hyphal growth and yeast form associated genes. Then specifically activates the so-called hyphal specific genes, including the ECE1. This is a gene that encodes the first toxin identified in Candida albicans, Candida lysine. Okay, and so to activate hyphal development. So you see that those technologies, ChIP-seq and RNA-seq, allow really to decipher the circuitry and explain at the molecular level what occurs in terms of regulation of the yeast to hyphal and transition. Of course, we confirm that by genetic interaction analysis to look at genetic interaction between the transcription factor and target genes, but also with protein-protein interaction analysis by looking at co-enrichment of some motifs of other transcription factors. And we found that, in fact, SFL1, SFL2 do not work alone in this regulatory circuitry, but work in collaboration with other transcription factors that regulate hyphal growth, EFG1 and DT80. Now, SFL1 and SFL2 are, as I mentioned, members of the HSF type DNA binding protein family in Candida albicans. That also includes the prototypical transcription factor and essential gene HSF1. This is a transcription factor that is highly conserved among different organisms, and it regulates the heat shock response. And on the heat shock response is a process that is very conserved among many organisms, including even mammals. And so this family also has another member called SKIN7. It carries a response regulator receiver domain, the so-called two-component signaling pathway. And it is initially known to be involved in protecting Candida albicans from oxidative stress. As you can see here, deletion of SKIN7 synthesizes, hypersensitizes Candida albicans to oxidative stress, so hydrogen peroxide. Now, introduction of a wild type copy restores, growth on oxidative stress conditions. Now, when you overexpress SKIN7, what we found, surprisingly, is that it activates hyphal development. So we wanted to look at how this occurs, and we performed genome-wide location and expression analysis to decipher the regulatory circuitry of skin 7 by tagging it with a triple HA epitope at the C terminus and placing it under the control of the tetracycline inducible promoter. And we found that actually, like SFL1, SFL2, skin 7 acts as both as activator and repressor of gene expression because it binds to the promoter and activates and represses at the same time the expression of target genes. So many transcription factors act at the same time as activators and repressors of gene expression. And we strikingly found that it binds to the promoter of many regulators of high-fold development, including EFG1, SFL1. And it comes somehow like SFL2, joining SFL2 in a similar manner by activating directly the expression of activators of high-fold growth, also a subset of the so-called high-fold specific genes, and repressing yeast form associated genes to, induce high-fold development. At the same time, it protects Candida albicans from oxidative stress. So now the missing guy is HSF1, and we have already in the literature the ChIP-seq data of HSF1, and we combined all of those data together from the group of Lee Cowan published in 2016. So you can see that by looking at all of the data together, we come up with this model in which those 4 HSF type regulators regulate in common this fascinating developmental process in Candida albicans, which is filamentous growth, as a feature of these transcription factors and their role in such a virulence threat is Candida albicans high-fold development. So by looking at this network more precisely, we notice this gene here, initially associated to the yeast form, genes, RME1. So RME1, actually in Saccharomyces cerevisiae, it is a transcription factor that regulates meiosis and sporulation through inhibiting the expression of a regulator IME1. In Candida albicans, RME1 is, as you can see here, is activated directly by SFL1 transcription factor. But we know that in Candida albicans, there is no meiosis, right? So what does RME1 do in Candida albicans? Does it regulate the yeast form growth as expected from this circuitry? So we did a ChIP-seq and RNA-seq analysis of transcription factor RME1, and we strikingly found that RME1 rather regulates chlamydospora formation based on the this transcriptional circuitry that we designed here. We found that many genes are directly regulated, upregulated by RME1 that are involved in chlamydospora formation. Chlamydospora formation markers, CSP1, CSP2, etc., cell wall genes, etc., but filamentation as well. And we know that chlamydospores are those thick-walled spores derived from a high-fold or pseudo-high-fold cells and required for growth under, harsh conditions. So it's not true that RME1 is finally associated with yeast form gene, but rather associated with chlamydospora formation. Okay, so we found this fascinating, and this allows to, like, initially when you look at the transcriptional circuitry, you could also go and see and expand the circuitry to further understand how morphological processes are regulated by transcription factors in Candida albicans and in other yeast species, if we could apply this strategy to other yeast species as well. Now, it's really great to see that sequencing and annotation of the Candida albicans genome allowed this creation of functional genomics resources such as gene deletion libraries and gene overexpression libraries, but also epitope tag and signature tag libraries. And with the development of those genomics technologies, allowing to create genome-wide profiling tool like next generation sequencing and algorithm for data analysis and data mining. Now we can combine multiple approaches, to answer to specific questions and infer gene function, discover pathways, and model biological networks. So by exploiting those loss of function, so-called loss of function screens and overexpression screens, or genetic perturbation of, let's say, a series of transcription factor deletion mutant or transcription factor overexpression mutant, you could perform high throughput phenotyping and combine to those screens, those genome-wide expression and location profiling to discover pathways, as I mentioned, and model biological networks. And so we created at Institut Pasteur, during my postdoc and later on by developing this tool, this first overexpression collection in Candida albicans that relies on a tetracycline inducible expression system I mentioned initially with Skin7. And we started with a pilot project, basically using around 600 strains, so covering 10% of the genome. That includes, each strain, overexpresses a given gene, including genes encoding transcription factors, kinases, phosphatases, DNA replication, recombination, and repair genes, as well as cell wall genes. So each strain carries basically two constructs, this collection, basically. So you have the gene to be overexpressed based on the control of tetracycline promoter, and also carrying a 20 base pair sequence or barcode, serving as a strain identifier and allowing to quantify the relative abundance of each mutant in mixed population experiments, right? And so the second construct is based on the ADH1 locus and consists of the overexpression of the reverse state activator, so allowing the system to work in Candida albicans, of course. So when you add doxycycline or tetracycline derivative, you induce gene expression in a strong manner. So you overexpress, basically. And so we constructed this set of strains up to 572 strain. And then we pooled those mutants together and we grow them, in the absence or presence of doxycycline. So you overexpress here, you don't do anything here as a control. Then you extract genomic DNA. You amplify the barcodes because you're surrounding each barcode with identical sequences here in every mutant, so allowing to amplification of all of the barcodes. Then you generate the library and you can perform bar-seq, so meaning high throughput sequencing of the barcodes to look at the relative abundance. So you count those barcodes reflective of the abundance of each mutant. So instead of performing the screen individually with different strains, you pool them, and with bar-seq, you could look at the abundance in one sample, in one culture, right? So I think we found this really powerful. So you could do this, of course, now with bar-seq, with high throughput sequencing, but initially we could do it with microarrays, right? And then the technology is substituting microarrays for, of course, high throughput sequencing. So we could normalize read counts and perform some calculation of relative abundance. And we found that, for instance, in vitro in a rich medium, the overexpression of certain genes has a fitness cost, right? For example, DNA damage response to cell cycle progression gene, when you overexpress them, has a fitness cost. So we detect them here in negative z-scores, but also those involved in developmental process like filamentous growth, you come up with SFL2, you come up with RME1 because it has a fitness cost, basically. We also applied this to biofilm formation in albicans. So biofilms are communities of highly adherent cells that are encased in a matrix and are somehow resistant to antimicrobial agents. And so we found a specific enrichment of many PGA genes. So those glycosylphosphatidylinositol linked proteins of the cell wall that contribute to forming biofilms in Candida albicans. And so these genes can act as adhesins, allowing to increase the adherence of cell between them in a biofilm, within a biofilm. Now, how to apply this fitness profiling, competitive fitness profiling, and phenotypic analysis in vivo in an animal model, for example, to look at the gastrointestinal colonization by Candida albicans. So you know that, as I mentioned initially, Candida albicans is a commensal of the gastrointestinal tract that coexist with other microbial components of the gut microbiome. But in a disease context, for instance, immune defects or mucosal damage, or in the case of treatment with large spectrum antibiotics, Candida albicans can increase its growth rate and can participate in disease severity or cause disease, basically, in the gastrointestinal tract. And so the question that we answered initially, we asked initially is, what are the genetic determinants of the ability of Candida albicans to colonize the mammalian gastrointestinal tract? So we initiated this screen in vivo in a mouse model of Candida albicans gastrointestinal colonization by taking bulb-C female mice, and we treat them with gentamicin and streptomycin to deplete the microbiota and facilitate gastrointestinal colonization. And then we provided to the drinking water doxycycline in one side and no doxycycline on the other side. This allows to overexpress or not those genes in the collection in vivo in a mouse. And so we allowed mice to the effect of the antibiotics for 4 days, and then we inoculated by gavage, oral gavage, the pool of strains together. And we allow them to propagate in the gastrointestinal tract for 10 days. Then we homogenize stools. So we get the stools and we homogenize them, and we perform total DNA extraction. We amplify the barcodes, and then we look at the relative abundance of each strain that overexpresses a given gene, and mice treated with doxycycline and control mice. And strikingly, we found one strain with increased abundance in a plus dox-treated mice versus unaltered abundance and minus in untreated mice. And this one gene out of the 579 genes is crazy2. So it's a transcription factor that is homologous to Candida albicans crazy1 and Saccharomyces cerevisiae crazy1. Crazy1 is involved in calcineurin signaling and calcium homeostasis in yeast and in Candida albicans, but crazy2 is not involved in this process. And it appears to have arisen through duplication of a common ancestor of crazy2 and crazy1, would be specific to the Candida lineage here. So we found this interesting, a gene whose overexpression, allows Candida albicans to better colonize the gastrointestinal tract in a mouse model of gut colonization. And so, and of course, appears to be Candida lineage specific gene. So what does it do? So we performed this combination of TRP-seq and RNA-seq analysis to look at the transcriptional circuitry of crazy2. And we found that among the 103 bound and upregulated genes, a subset of mannose-transferase genes, cell wall, cell surface genes, some of the transcription factors also are bound and activated, etc. On the other hand, we found 63 bound and downregulated targets. So here again, crazy-2 acts as activator and repressor of gene expression. So based on those findings, we have here a bunch of mannocell-transferase genes and cell wall genes as well. So we might be dealing with protein glycosylation here, right? And production of cell wall proteins. So we wanted to test this hypothesis later on, but before we looked at the transcript profiling data and we performed some gene set enrichment analysis of the transcript profiling data. And we found that actually among the upregulated genes, we have a strong enrichment of genes linked to hypoxia. On the other hand, among the downregulated genes, we found a strong enrichment of genes pertaining to aerobic respiration. So this is telling us that here, when you overexpress crazy-2, there is a change in the transcriptional program reflective of an adaptation to a oxygen-deprived environment, right? Hypoxia. So this is really exciting, and we wanted to test the hypothesis that this gene adapts to a hypoxic environment, make Candida albicans adapting to hypoxic environment, and it might somehow regulate processes linked to N-glycosylation or cell wall dynamics while it colonizes the gastrointestinal tract of mice. And we tested tunicamycin, the effect of tunicamycin, which is an inhibitor of N-glycosylation. And we strikingly found that the overexpression of crazy-2 hypersensitizes Candida albicans to tunicamycin, specifically at 37 degrees Celsius under hypoxia, but not in normoxia at 30 degrees Celsius. So you see here the validation of our assumption based on the data we acquired from deciphering the regulatory network of crazy-2 and the gene set enrichment analysis that we performed initially. And so this is really specific to tunicamycin, but not additional drugs that could have an impact on unfolded protein response or et cetera. So there is something clearly happening here with regard to cell wall and glycoprotein production, while Candida colonizes the gastrointestinal tract in a somehow environment that is hypoxic or mainly hypoxic. And so this is our working model where we believe that crazy-2 is somehow a transcription factor that allows Candida albicans to better colonize the gastrointestinal tract that responds to hypoxic condition at 37 degrees Celsius and directly activates mannocell transferases, cell wall or cell surface genes and hypoxia genes, while repressing directly cellular respiration to somehow modulate glycoprotein production and GI tract colonization. So I hope in this part, I convince you that studying the function of transcription factors, which are decision makers in living cells, allow to identify molecular mechanisms that underlie the control of pathogenic and commensal threats of microorganisms, but also that the systematic genetic perturbation of transcription factors combined to functional genomics approaches like TRIP-seq, RNA-seq, and bioinformatics allows to map the regulatory networks and infer transcription factor function, so the so-called functional inference that we talked about initially in this talk. And that the availability of signature tagged and genetic resources in Candida albicans allows to implement those powerful competitive phenotypic assays to study both in vitro and in vivo pathways and circuitries. And so I hope I convince you also that Candida albicans expresses pathogenicity and commensal threats by activating specific transcription factors like SFL2, SCN7, crazy-2, And this way, converting environmental signals like temperature, oxidative stress, and hypoxia into adaptive threats in different niches of the mammalian body. I don't know what time can I go for like 15 minutes? Yeah. Okay. What time is it already here? Okay. So I'm going to be providing you with some recent data and recent project dealing with a facet that is more about forward genetics here rather than reverse genetics. So we're looking at natural forward genetics as collection of clinical isolates, Candida albicans, that are adapting to condition of the host and seeing the genetic determinants of those phenotype developing in the host. So, as I mentioned, Candida albicans, colonizes the oral cavity, the gut, and the vagina in healthy individuals. But under some disease context, Candida albicans colonization can increase and can contribute to a disease severity. And this is the case of cystic fibrosis. So in cystic fibrosis, Candida albicans in healthy individuals, it's not supposed to be colonizing the lung, but in patients with cystic fibrosis, Candida albicans somehow translocates to the lungs and participate to disease severity. And this has been demonstrated in many publications recently. And so the idea here is to look at the genetic determinants of Candida albicans' ability to thrive within the airways of patients with cystic fibrosis. So patients with cystic fibrosis have this CFTR mutation that makes mucus in the airways and serves as somehow an incubator of microbial organisms. So we initiated this project with clinicians from the Children's Hospital in Tunis, in Tunisia, by recruiting 25 cystic fibrosis patients, including 2 adults and 23 pediatric patients. So those are antifungal naive, that they have never been exposed to antifungal agents. Sampling occurred for 2.5 years, and we collected out of the airways of those patients 146 Candida albicans clinical isolates. And when you look at the population structure of those isolates, so we look at the diversity really, but more than half of them are colonized with strains from a specific clade, clade 4. And clade 4, shown here, is a clade that is enriched in strains from Middle East Africa. So you know that Candida is intimately linked to a human body, and so it co-evolved with the human body. So we have clades that could be associated to some geography also. And we found that each patient, basically during time, is colonized with strains sharing the same DST, diploid sequencer. So they are genetically highly related, and maybe one single strain is colonizing, the airways of those patients. And one patient in particular, this patient with the code HBJ, was sequentially sampled over a period of 1 year. This patient is colonized with a strain from clade 10 in red here, and we were able to collect 18 isolates from the airways of that patient. So we look at those 18 isolates. Those are those that initially were isolated, and those that over time, the more recently isolated, right? Within 1 year period. And so we look at the growth of those Candida albicans and colony morphology features, and we strikingly observe this fascinating, phenotype, like fuzziness of a colony. So you have correlating with filamentous growth. So strain HBJ61 here and strain HBJ63 here are fuzzy in terms of colony. And this correlates with filamentous. So we can say they are hyperfilamentous, basically, right? Compared to the remaining isolates. And so we found that this is also the case a bit for another strain, 4.3 here. See here in RPMI, it appears hyperfilamentous compared to the most of the remaining isolates. And we know that filamentous growth is is strongly linked to the ability to form biofilms as well. So we tested the ability of those strains to make biofilms. And we found that the increased fuzziness and increased filamentous growth correlates with increased ability to form biofilms. 6.1, 6.3, and 4.3, but not 6.2. 6.2 from the same sampling, that we got from the that patient. And so we know that Candida albicans is exposed to CF-associated pathogen in the airways of those patients. And so we might think that they can make a dual species biofilm with those CF-associated microbes, including Pseudomonas aeruginosa or Staphylococcus aureus as well. And so this work by Rebecca Hall, show this nice picture of a Candida albicans that forms mixed species biofilm with Pseudomonas aeruginosa, as shown here, and say that they do that by protocol by co-incubation of Candida albicans with Pseudomonas and DMEM medium, and then we can look at the survival CFU counting of Candida albicans, within those mixed species biofilm, and we strikingly found that those hyperfilamentous and strong biofilm formers have the ability to better survive in dual species biofilms formed with Pseudomonas aeruginosa. 6.3, 6.1, 4.3, but not 6.2. The weakly, the strain that forms, that weakly forms filaments. So what is the genetic determinant of that feature? We sequenced the whole genomes of the complete set of strains. And we did this in collaboration with Corinne Maufray, a talented bioinformaticist, and with the Biomix Core facility at Institut Pasteur, Imène Najor and Laurence Ma. And as you can see here, this is a density of heterozygous SNPs displayed by each strain. You can see here that the strains are highly homologous, highly, genetically close to each other, forming somehow a clonal subpopulation structure. And so we mined those data by looking at the SNPs displayed by those hyperfilamentous and hyperbiofilm former strains compared to the remaining isolates. We found 35 non-synonymous heterozygous SNPs in 19 ORFs, differentiating those hyperfilamentous strains from the remaining isolates. 13 of those 35 are found only in 7 ORFs and are not present in a set of 182 isolates from non-cystic fibrosis patients, published by this nice paper where they looked at the genome of 182 isolates from patients and from healthy individuals as well, but not cystic fibrosis patients. So we found this set of ORFs very interesting, as it might include maybe the genetic determinant of this threat linked to cystic fibrosis. And we found particularly this gene showing up here, ROB1, a transcription factor of the zinc cluster family of regulators that is a master regulator of biofilm formation in Candida albicans. And we specifically found it carries a SNP consistent of a glycine, substitution of a glycine at position 299 by glutamate. And ROB1 is really like a member of an intricate circuitry of transcription factors that control biofilm formation in Candida albicans. And we think that this mutation acts in a similar manner to mutations that you could detect in so-called zinc cluster family of regulators that are prone to these mutations, and this mutation allows those transcription factors to to be transformed into a hyperactive form, like somehow a gain of function mutation. And this is seen in transcription factors that regulate drug resistance in Candida albicans, such as TAC1, UPC2, MRR1. So we think this is the same process. like the strains here have acquired a mutation in transcription factor that is critical for their ability to form biofilms, to hyperfilament, and maybe better interact with Pseudomonas aeruginosa in a cystic fibrosis context. So introduce this mutation in the hypofilamentic or non-filament, weakly filamentous HBG62. We strikingly found that it becomes hyperfilamentous. We looked at the expression of genes, target genes, the hyphal specific genes. Look, highly increased in mutant, not in non-mutated cells. We look at biofilm formation. We strikingly found a high level of biofilm formation on polystyrene plates. And we looked at the survival of those mutants in dual species biofilm. And we found that indeed ROB1G299E enhances Candida albicans survival in dual species biofilm formed with Pseudomonas aeruginosa. So we pinpointed here a SNP and mutation that likely has arisen through while the Candida albicans was colonizing the CF airways of that patient, allowing it to better compete with Pseudomonas in biofilms. But this has been already shown elegantly by a study from Leah Cowan's lab in 2015, showing that actually, Candida albicans in a cystic fibrosis airways can adapt to this context by evolving hyperfilamentous strains. And those hyperfilamentous strains, are resistant to the filamentation repressing effect of Pseudomonas aeruginosa through a mechanism involving loss of function mutation in a repressor this time called NRG1. We talked about NRG1 initially in my talk, but this is really an elegant study that initially, was directing us towards this pathway as really like regulators of hyphal development that acquire mutations, allowing Candida to be hyperfilamentous, and this way allowing it to resist to the filamentation repressing effect of the microbes surrounding it. And so we tested this hypothesis, of course, with our strains. The strain that carries the ROB1 gain of function mutation is not inhibited. Its filamentation is not inhibited by Pseudomonas. Although it is inhibited, so the pseudohyphal growth of HBG62 is inhibited when you expose to Pseudomonas here, but not here. And so we know that Pseudomonas, releases phenazines, right? So these are the quorum sensing molecules that, inhibit filamentous growth in Candida albicans. And so by exposing Candida albicans, the mutant that we constructed, to a phenazine analog called PMS here, you could see that the filamentation is not repressed anymore, right, in the mutant, whereas it is repressed a sufficient amount in the wild type strain. So I hope I convinced you that this is really also somehow another mechanisms allowing Candida albicans to adapt to a pathological context, which is cystic fibrosis in the airways of those patients, by not loss of function mutation in the repressor, initially and elegantly showed by Leah Cowan's lab in Toronto, but by also gain of function mutation in ROB1 that allows Candida to hyperfilament, but also to somehow survive better in biofilms that are formed with Pseudomonas aeruginosa. So I thank you for your attention. I'm sorry to be a bit taking a lot of your time more, but I hope you enjoyed the talk, and I thank you very much for listening to me. And thank you all.
Thank you, Sadri. Very exciting science. Any question from the attendee? I have one for you, Sadri.
Yes.
just curious about the function of crazy 2. So when you overexpress it, you have in the presence of glycosylation inhibitor. So the strain is, I mean, have some defect, right?
no defect, yeah.
Is there any clue? Do you have any clue on why you have seen this?
So the thing here is that you have a apparently high level of N-glycosylation occurring there. So the effect of tunicamycin is more pronounced, right? So you would increase the susceptibility, whereas in a knockout, you would see that there's a more of a resistance to tunicamycin is shown here. And now, what is the link of protein production or somehow N-glycosylation process with that? So we've tested the mutant, overexpression mutant with Dr. Jouot. It's in Lille. Basically, we did some analysis on the capacity of the mutant to like somehow adhere more to explant of the colon, right? We incubated Candida albicans overexpressor and deletion strain and co-incubated it with some of the gut explants here. And we washed and we look at the CFU. So apparently what happens here is that the adherence, like the strains that overexpress crazy2 appear to be more adherent to the colon explants than the controlled strain, right? So there might be somehow a increased expression of some adhesion at the surface through N-glycosylation process of glycoprotein production that are adhesins, and could maybe stick better to the colon tissue. This has been obtained, right after publishing this paper. Could be a nice addition to the paper to look further highlight on the function of crazy2 and there. Yeah. So thanks for this. This
thank you, Saidi. Any question? Other question? Come on, guys.
Yeah, it might be a lot of information that is provided at the same time, and maybe, but I try to facilitate a bit with more didactic approach for students, as I know that this is a training, network. So, yeah, I try to be
definitely, and you'll meet with fellows, so maybe you can get more questions directly. So another question, Saidi, related to the crazy2. So if I understood, overexpression crazy2 we're suspecting you have more manozylation activity, right? Mm-hmm. Is there any effect on overexpressing crazy2 on the either M-masking or masking phenotype of the cell wall? So in other words, like overexpressing crazy2 might have more manosylated protein, or
Yeah, definitely. This is something we need to understand maybe in the future, looking at the manosylated profile. But what we did was a gel shift, the so-called, I have a lot of data here showing so this is the HAX1 electrophoretic mobility shift assay. When you could look at the N-acetyl hexosaminidase activity, and the more it is, I think can't remember actually the experiment that we did initially. Yeah, exactly. So you have a difference in the in how the HAX1 protein migrates, so making reflective of the glycosylation pattern that has changed, basically, and you could see it clearly here. do not see it that perfectly with the knockout, but you see it clearly that there is a shift in distribution of HEX1 activity right in the gel. So suggesting that most likely we have something that occurred with regard to the glycosylation pattern of a protein. Now, is HEX1 a target of? But this is something that we see as a marker of glycosylation, perturbation of glycosylation, basically in a cell that is specific to crazy 2 overexpression. We don't see it in a control strain and another wild type strain. You see a bit of shift from the other side, knockout, but not that perfect. Yeah, it's not clear, but we need maybe quantitative.
Yeah, I think it's maybe worth quantifying, like Manon, for example, at the cell wall by fax or something like that.
That's right. That's right. Yeah.
We have a question from Sarah.
Sarah, yes, Sarah.
Hello, can you hear me?
Hello, Sarah.
Yes.
Yeah, we hear you.
Hi, great talk. I had a quick question, just following up on the CRZ2 pipeline. I thought it was really interesting. So I've done a little bit of work with CRZ1, and I thought it was really interesting how their functions are like completely different from each other. And then you briefly spoke about the CRZ2 overexpression and how it leads to increased adhesion. So I was wondering if you've looked at biofilm formation with CRZ2 overexpression and what you think that would look like.
Yeah, we did not test biofilm formation, although we know that Candida might, develop into biofilms on both abiotic but also biotic surfaces, including, mucosa of epithelial cells or, intestinal cells. What kind of biofilm it forms basically is really a question that would definitely looking at in vitro biofilm development on polystyrene plates or on a microfermenter versus those that form in between microorganisms. I show Pseudomonas can do it with Candida, with staph, I believe, but in the context of a tissue and how microcolonies followed by maybe biofilm development could occur on a tissue is something that we did not explore yet. But your question is definitely important. Look, is adherence followed by better microcolony formation and biofilm formations? That's what you might be expecting. I'm eager to know, and thanks for pointing to this experiment as a potential to explore.
No, I was just curious because oftentimes we see that adhesion is often linked to robust biofilm formation. So I was curious if like an overexpression compared to the wild type compared to deletion mutant.
The thing, Sarah, is that we would expect to see some of the filamentous growth, but because filamentous growth is also linked to biofilm formation as well. If you have a mutant that is defective in filamentous growth, usually you not expect to see that level of biofilm formation. Although this is not necessarily true, because you can have some Candida species that are not filamentous and still they can form biofilms, of course. But for instance, we did not detect any filamentous growth when you overexpress crazy-2. At least in the condition that we tested, meaning YPD or some filamentous,
Good, thanks, Saidi. Any other question? Just one quick question, Saidi. I just saw, my interest in sulfur metabolism in Candida. So I've seen, like, I think I was overexpressing crazy-2 have some upregulated genes in the overexpression mutant related to sulfur metabolism. So that is seen in your GCA. And have you tested any?
No, we Yeah, we did not test. I just want to maybe display and then gene set enrichment analysis here. And so sulfur and amino acid metabolic are linked basically, and it's not that much of a as you can see, of enrichment. Like circles are a bit tiny, but I did not test if sulfur compound metabolism could be affected by genetically perturbing crazy 2. This is something that definitely we need to do some of the maybe metabolomic approach to look at the metabolites that could have, maybe would explain the behavior of those cells, of those, mutant, both overexpression and deletion, because you would expect reverse things happen, although not necessarily. Yeah, but yeah, I mean, some of the categories are really like oxidative is popping in there. So they see it also in what's it there? So
Yeah, it could be
it could be
an indirect effect.
indirect
because
effect. Yeah,
sulfur metabolism usually is also related to oxidative stress response.
Stress. Yeah. Okay.
As glutathionein is required for that. Okay.
Yeah.
Thank you, Sadri.
You're welcome.
I think if there's no other questions, I'll leave. Thank you so much, Sadri, for accepting the invitation.
You are welcome. I'm really happy to still discuss with you. If you have any also for students, if you need to reach out to me, I'm
yes, I
happy
think
to
you
answer.
have. I think Maria-Hélène will basically drive like short get together with students. So I'll leave you with them. So thank you again, and
thanks, Adnan.
Happy to see you in Canada again.
Thank you very much.
Okay, take care.
Bye.
Thank you.
Take care.
Merci, Adnan. Okay, I'll stop the recording, guys, and